Hi, my name is Olurotimi Meshubi. I'm an assistant professor in the Division of Cardiology here at the Johns Hopkins University School of Medicine. I am a physician scientist and an electrophysiologist, and I'm delighted to present the highlights of our recently accepted manuscript to the Journal of Clinical Investigation. I am the lead author on this paper, and I'd like to acknowledge all the contribution of all the collaborators from different institutions uh, towards this manuscript. I'd like to highlight first my mentor and the principal investigator on this study, who's Dr. Mark Anderson, the William Osler Professor of Medicine and the director of the Department of Medicine here at the Johns Hopkins University School of Medicine. In addition, I'd like to highlight Dr. Natasha Zakara, who's a glycobiology expert, as well as Dr. Jerry Hart, another glycobiology expert, both here at the Department of Biological Sciences in the Johns Hopkins University School of Medicine. Dr. Hart is the discoverer of a protein modification known as O-GlUCNAC modification that I'll be referring to and form a critical part of our study. Atrial fibrillation and diabetes are both huge global public health problems that cause increased mortality and morbidity. It's well known that diabetes is an independent risk factor for atrial fibrillation. However, we do not have a good understanding as to how diabetes causes atrial fibrillation. Our group studied a protein kinase known as CAM kinase, which has been well described to be involved in causing pathology in various cardiac conditions such as heart failure and various arrhythmias such as ventricular arrhythmias and atrial fibrillation. So, to tell you a little bit about CAM kinase, here's a cartoon showing CAM kinase as a monomer. It exists typically as a conglomeration of 12 subunits, but here we're showing just a monomer. The monomer consists of three domains. The first is the association domain, which is highlighted here in green, through which it binds to other monomers to form this 12-unit protein. And next is the regulatory domain, which is highlighted in purple, which contains various amino acids that can be post-translationally modified to affect the activity of the kinase. And lastly, in orange, which is the catalytic domain, which typically covers the regulatory domain in the resting state to keep the kinase in an autoinhibitory state. However, in the presence of calcium and calmodulin, undergoes a conformational change where the catalytic domain opens up as is shown in the middle panel in this figure and thus becomes active. Subsequently, the various sites for post-transcriptional modification in this regulatory domain then get post-transcriptionally modified and can keep CAM kinase in this active state even in the absence of calcium and calmodulin. Now, if you focus down on this regulatory domain, as highlighted in this slide, you see the various post-translational modifications that have been described. Highlighted here as the post-translational modification caused by oxidation, which was first described by our group, and it causes oxidation of two methionine residues that appear next to each other at the 281 and 282 site. Right adjacent to that is a serine residue at the 280 site that has also been described to undergo a different post-translational modification known as O-GlUCNAC modification, where a single N-acetyl glucosamine residue is added to modify the protein. So for the purposes of this study, we were interested in finding out if CAM kinase contributes to or is critical for promoting atrial fibrillation in diabetes. And this was based on our previous studies where we've been able to show that CAM kinase is a driver of atrial fibrillation in the context of hypertension and also in a mouse angiotensin 2 infusion model in the mouse model. So to answer these questions, we utilized two diabetic mouse models, the type 1 and the type 2 model. For the type 1 model, we used the high-dose streptozotocin model, where for the type 2 model, we used a combination of high-fat diets as well as low-dose streptozotocin injections. And we utilized a portfolio of various genetically modified mice either that were previously created in our lab or that we newly created for this study, where we could toggle various post-transfusion modifications, specifically oxidation and Okooknack modification, to see what the effects were in the context of diabetes to drive atrial fibrillation. Now I'd like to summarize or highlight some of our findings. So first, we were able to establish that CAM kinase is a critical driver of atrial fibrillation. You can see from the slides, wild-type diabetic mice were prone to atrial fibrillation in the context of diabetes, whereas mice expressing a protein known as AC3I, which is a CAM kinase inhibitory peptide that is overexpressed in the heart, protected these mice from atrial fibrillation. Having established this, we're interested to find out what specific post-transfusion modification, in this case oxidation or Okooknack modification, a combination of both, was responsible for the role of CAM kinase driving atrial fibrillation in diabetes. So to achieve this, we used a couple of mice. So there's the MMVV mice, which is a mouse in which we replace the methionine residues by valine, and these mice are resistant to oxidation. And these mice were protected from atrial fibrillation in the presence of diabetes, both in type 1 and type 2 models, suggesting that oxidation of CAM kinase was a critical driver of atrial fibrillation in this model. Next, we created a new mouse model based on the previously described modification at the S280 site, where we replaced the serine with an aniline, thereby preventing Okooknack modification at this site. And to our surprise, these mice were still susceptible to atrial fibrillation, suggesting that Okooknack modification at this site did not play any role in preventing atrial fibrillation. To dig into this further, we tried to assess for evidence of Okooknack modification at this site, and again, to our surprise, we were not able to identify Okooknack modification by mass spec, looking at various diabetic hearts for this modification. Thus, based on these findings, we decided to take a step back and see if specific targeting of Okooknack modification at a more global level played a role in this phenotype that we observed. So we generated a new mouse which expressed a particular protein or enzyme known as O-gluconaccase or OGA. This is an enzyme that prevents O-gluconac modification of proteins and thus would prevent O-gluconac modification from taking place in whatever tissue. So we specifically overexpressed this in mouse hearts, and these mice were protected from atrial fibrillation both in type 1 and in type 2. And thus this was an unexpected finding, but I think opens up a whole new area of interest and investigation. To summarize our findings, we were able to establish that CAM kinase is a critical sensor for atrial fibrillation in diabetes, and specifically this is through oxidative post-transfusion modification and activation of CAM kinase. We did not find any evidence for O-gluconac modification of CAM kinase or a role for O-gluconac CAM kinase in these our studies. However, interestingly, we found that global O-gluconac modification plays a role in atrial fibrillation. However, we did not go further to figure out which specific downstream targets were being modified by O-gluconac to effect these changes that we observed. We believe that this will form a whole new interesting area to go back to explore and study to better understand what role O-gluconac plays in atrial fibrillation. Thank you very much, and I hope that you enjoy reading our study.